Thank you very much, Alan. Can you all hear me okay? Can you all see me okay? So, as you'll be aware from what Alan said, this is where we both work. This is the Institute of Psychiatry. And has anyone been there from here? Yeah. Well, you'll all realise then that that's the only tree in South London. And we're very proud of it. So who here thinks that there's been some significant advances in our pharmacological treatments of psychiatric disorders over the last 10 years? Okay. Can anyone name me a highly effective new compound that's come in to psychiatry in the last 10 years? Better than something we had available before. Revolutionise your practice. So I've been doing a bit of a literature search for you, and this is what I found. I mean, that's where we're at, I think. I don't think we've really made many advances at all, pharmacologically at least, in our treatment of psychiatric disorders, and particularly amongst the neurodevelopmental disorders. So why is that? I think there's a number of cultural reasons and a number of scientific reasons. And firstly, I'm going to go through some of the sort of cultural issues about the way we work, and then I will talk about some of the scientific issues generally, before focusing a little bit down on autism. So, for those of you who don't know, this is the elephant in the room, and these are the things that we're not allowed to speak about. So, one of the barriers to us making progress is the fact that actually a lot of us don't get on very well with our basic science colleagues, because we talk a different language, and they quite rightly think we're a little bit intellectually impaired when it comes to neurons. So, I mean, for example, does anyone know that terrible joke about, Frank, apart from you, what the definition of a neuropsychiatrist is? It's a psychiatrist with one neuron, all right? So that's kind of what they think of us. And then, of course, we don't help ourselves. Sometimes this, I think actually this represents a surgeon, of course. There's how we get on with some of our scientific colleagues. So we can be some difficult colleagues to work with as we think we know best. And then there's this other issue, right, about what the general public think about us. So these are the Scientologists. They actually have a museum called Psychiatry, the Museum of Death. So lots of people don't appreciate what we do. So there are these socio-cultural reasons about the way we work with each other and the way we work with the society that really impairs our ability to make progress. The other impairments to progress are the way we think and the way we work. So are most people in here adult psychiatrists? Yeah? Any child psychiatrists in here? Yeah. Do you run any services, joint services with adult psychiatrists? No, no. So classically what we'll do, of course, right, is we will be doing this, is that we will be trying to treat something once it's started. And the further we go into adult psychiatry, the further down here we get. And it has to be said that it's very rare to find, say, a depression service that cuts across ages, or a psychosis service that cuts across ages, or an ADHD or autism service that cuts across ages. And so we tend to come in very late, and we tend to come in with our own way of looking at things. And of course, what we want to be doing, if possible, is getting up as close to this as we can to stop this boulder rolling down the hill. The other scientific bar to our progress is the fact that we're dealing with heterogeneous disorders, biologically heterogeneous and clinically heterogeneous. So what I mean by this, so this is obviously an Andy Warhol. These are different tins of soup. They all look identical on the outside. But within those tins, you can't see this unfortunately very easily, there's different flavors of soup. So for example, you'll be treating someone with psychosis, but within that group of individuals with psychosis, there'll be many different biological or cognitive subtypes. The same with depression, the same with autism, the same with ADHD. So what we need to be doing is using much more personalized medicines approaches so that we're aiming for the person in that tin. We want to be getting our treatment into that person. We don't want to be treating 100 people with the same treatment to find that just 1 or 2 or 3 recover. So how do we go about doing that seemingly impossible task? So now I'm going to give you an example in this next part of the talk on what we're doing within autism. And believe me, if we can make progress in autism, you can make progress in virtually anything. And so I'm going to give you an overview firstly on what autism is, and then I'll talk you through some of the scientific thinking about how I'm trying to make some progress. So it affects about 1 in 88 of the population. It's 4 times more frequent in men than in women. We don't understand why that is, but we think that the male brain may be a risk factor for developing autism. So when you get the disorder superimposed in a male brain, you're much more vulnerable to expressing the disorder. So 1 in 88, 4 times more common in guys than girls. It's very, very, very genetic. It's highly genetic. The trouble is, we don't know which specific genes are causative. We know that there are families of genes which come in particular molecular pathways which increase risk. And we know there are these things called CNVs, copy number variants, which very significantly increase your risk. We know that there's essentially 3 major neurochemical candidate pathways, not the only ones, but the major ones that most people are focusing on. And that's the serotonergic system, the glutamatergic GABAergic system, and neuropeptides in terms of oxytocin and vasopressin. So even if you're not interested in autism per se, I'd say you need to be interested in it as a practicing physician because you've got very, very high rates of depression and anxiety of about 75% in young males, very high rates of epilepsy, very high rates of ADHD, and you've got a very significant reduction in lifespan. So I don't know if any of you saw that paper in the Yellow Journal a couple of months ago or so. If you're of a lower IQ and you have autism, you're going to die about 20 or 30 years younger than someone who doesn't have autism. And if you have a regular IQ and you have autism, you're going to die about 15 years younger, mostly through an increased risk of suicide or seizure. So what treatments do we have for the disorder, the core disorder? The answer goes back to that blank slide. We have none. We have no highly effective treatments for the core symptoms. We do have treatments for the associated symptoms, such as irritability and sleep, and depression, anxiety, etc., but none for the core symptoms. How are we doing then in terms of the treatments that we currently use within autism? So this just compares the pharmacologic approaches across all the countries that we could find. So this goes here from, this is Brazil, Mexico, Canada, USA, Japan, UK, Spain, Italy, Germany, and France. And this is the treatments that are used beneath the age of 19, and this is the treatments that are used above the age of 19. So can anybody see a pattern in there? Each colour, by the way, represents a different kind of medication. Can anyone see a pattern in there? No, right? There's no pattern. There's absolutely no pattern. So unless you're going to convince me that the clinical phenotype of autism varies so much across these different countries, clearly we've got idiosyncratic prescribing habits going on, using medications for which there is no evidence base. So we need to do better. So how are we going to do better? So this is the approach we took and we're taking now. We're saying, look, there are all these loads of genes, but we think they map onto a much smaller number of molecular pathways because they kind of come in gene families, which in turn map onto a smaller number of biological pathways, which in turn map onto a smaller number of brain regions and circuits to give you the clinical phenotype. So that's the way we're doing it, in a slightly reductionist way. But the most important way we're doing it is we're saying that we need to work together across Europe, and we need to bring basic scientists together with clinician scientists so that our work in rodent models can be directly translated into humans and back in parallel work, so there's no delay. So what classically would happen would be, say that, Alan publishes something in depression, I'd read it, I think that's great, and then I would try and do something, but Alan's already done that work 4 years ago, right? And now he's on to something else. And now I'm taking up that work. It takes me about 7 years or so to get that going. So that's a huge delay. What we want is contemporaneous work going across these different themes of work, from cellular assays, that's iPSCs, animal models, translate that from rodent to human, and then run clinical trials. And so that's the approach we took, number one. And the second approach we took is we want to get that boulder, we want to stop that boulder rolling down the hill. And so we're carrying out longitudinal follow-up studies in 1,000 people, starting at infancy, going into childhood and then into adulthood. And we're then also looking at people who've got very high risk genes for having the disorder. And what we're trying to do is to see, are there any biosignatures that we can use to bring these disparate groups of people into more biologically homogeneous groups, and therefore get a biological signal that we can hit. And obviously we'd be using the rodent models and iPSCs for that. So that's the general background. In the next bit of the talk, what I'm going to be saying is, what progress have we made and what are we finding so far? So the first thing we said was, okay, there are these risk gene things going on, these CNVs. We've got to find out what makes those more common. What makes you more likely to have that CNV? Then we've said, well, what does that CNV do to your brain? And then we've said, well, does that map onto what we know about autism? And then we've said, well, can we do anything to reverse that? And can we work together in human and rodent models to reverse that? And the examples I'm going to give today are trying to reverse it by targeting serotonin and glutamate GABA. And then, can we take that early experimental work and work together with our colleagues who are the regulators, the FDA and the EMA, for them to say, yeah, we agree with what you're doing, you can take that into trial. So, what's one of the main risk factors for you expressing a deleterious CNV? So, how many ladies are there? Oh, yeah, okay. Ladies, anyone here thinking of leaving your partner at the minute? Okay. Because the take-home message from this slide is, if you are, you've really got to go for a younger guy, okay? Because if you are going to have more children, the risk of your child expressing a risk CNV is very significantly related to the age of your partner, okay? So all that stuff you don't want to say out too loud is absolutely true. Go for the younger guy. So we found that, number one. And then what we found was, when we look in another group in that population, we take that risk CNV, we find in the healthy population who carry this CNV that don't express the disorder, they've got very significant differences in the anatomy of brain regions which are implicated both in psychosis and in autism. We then go on and we say, how closely do those map on? And is it related to the degree of symptomatology? And the answer is yes, it maps on very closely, and it is related to the degree of symptomatology. And this is in regional brain volume, okay? But that takes us sort of everywhere and nowhere. Remember that song, Everywhere and Nowhere, Baby? Right? We've got these volume differences that are associated with the disorder, but I'm going to suggest to you that I think that's a terrible target for a treatment. I don't think I'm going to be able to reverse that brain biology quickly with a pharmacological agent, number one. But number two, it can be mildly useless, because if I describe a volume difference to you, you can see these two things have the same volume, but they're clearly very different things, right? And there's another thing that that volume thing doesn't do. It's not necessarily very clinically useful, and I'll show you what I mean. So Claire, come here for a second. So for those who don't know, this is Dr. Capstick, a fine physician. So Claire's going to stand next to me, okay? We're about the same volume, agree? Right. Right. Now, which of us is a woman? Hands up for Claire. Fantastic. You're not sure, sir? Medical degree, sir. She's a woman. Okay, well done, Claire. Thank you very much. So, how did you decide that Claire was a woman? You looked at her, right? You looked at her. Right. So now, if you're a psychiatrist and you had to diagnose Claire as a woman, you would come up with the DSM-5 Claire schedule, right? Which would be about that tall and, 3 volumes probably. You'd have committees to go through it, and you'd ask such difficult questions as, well, what do you think? Do you think we should have, I like shopping as a diagnostic question? I weep in Love Actually. Is that another diagnostic question? I love cooking. Is that another diagnostic question? Well, you see, if you use those as diagnostic questions, I would score positively on all those, right? I weep like a sucker in those movies, love cooking, I'm clothes obsessed, all that kind of stuff, right? So, completely useless. Why don't we just do what you do and use that biology and see how good the machine is at categorizing people, whether they have autism or not? And if we do that, we can get an accuracy of about 90%. Now, let me be very clear. I am not saying you would therefore use this as a diagnostic instrument. What I am saying is that we can use this biology to begin to categorize individuals into subtypes of syndromes, and in this particular example, autistic or not, without looking at symptomatology alone. So this biomarker information begins to have proof of concept that we may be able to use it to stratify individuals as to having disorder or parts of the disorder. But again, what we need to do is make progress in treating symptoms, right? And some of the core symptoms of autism are abnormalities in social functioning, repetitive behaviors, and inability to switch your behaviors. And one of these people is socially abnormal. I'll let you guess which one. And so what we've been doing is saying, how do we unpick these really complex social constructs? And so we've been looking at how is it that individuals process facial emotions in an unconscious way? How is it that individuals respond to human versus non-human sounds? And how is it that individuals switch their behaviors depending upon which kind of human or non-human sounds they hear, or depending upon what sorts of social cues they get from the environment? So that's in adults. And this is when you'll begin to think, I'm on the wacky-backy. Remember pushing that boulder up the hill. We're also beginning to ask very similar questions in infants who are at risk of developing autism. And I'll now take you through the adult work first. So what we've demonstrated firstly is that when individuals with autism who are young adults do a social task or an inhibition task or whatever it is, they have significant differences in brain function from controls. But then when we modify their brain serotonin, when we reduce their brain serotonin, we abolish case control differences. So at the top here, you see case versus control differences. In this particular example, it's in a response inhibition task, but we get the same in a social task, whether it's a visual task or a sound task. So we get these functional differences that when we deplete the brain of serotonin, we abolish. But we depleted that brain of serotonin by using a thing called acute tryptophan depletion. And I don't know if any of you have ever tried that. I mean, it's essentially giving people a ghastly drink. We try and disguise the ghastly drink with a mint flavor, and so you end up with a mint-flavored ghastly drink. And it's just not going to translate into a treatment in the clinic. So this is just proof of concept that we can modify brain function. So then any ideas how we get that into the clinic, or might get that into the clinic? Well, I've put this slide in especially for Alan, because a few years ago, Alan said to me, well, haven't you ever thought of using this stuff called tianeptine, which is a SSRI, to also try and deplete the brain of serotonin? So an SSRI would increase brain serotonin. SSRI should be reducing brain serotonin. And of course, being a good and honest young man, I said to Alan, what's that stuff? It turns out made in France, etc., etc. And so we thought, well, if we really want to translate this into the clinic, we need to be using that approach. So we repeated this study with tianeptine, and we get exactly the same result. So having done that, we're now at a stage, I'll just show you the data if you really want to see it. So this here is under placebo. This is people with autism. This is the control here. Sorry, this is the control here. And then what we do is we show at baseline, and then we treat with tianeptine, and we take the ASD up towards a control level. So always look here, so here and here. Then we take the ASD up to control level after tianeptine, and ASD to control level after tianeptine. So what that demonstrates is proof of concept that we can use this medication that might work in a translatable clinical situation to modify brain function and therefore modify symptomatology. So the next step for us then is to take that into the clinic, but it's not to do the following. It is not to treat all comers. It's to specifically treat individuals who've got hyperserotonemia, who've got raised blood serotonin, and who we find at baseline we alter brain functional differences. So that's how we're going to enrich our sample. I've no idea whether it'll work or not yet, but I can tell you what wouldn't work, and that would be if I run a trial with 100 individuals with ASD within it. I'd expect I might get 15 or 20 responders, and that would be absolutely undetectable using regular clinical trials sort of techniques. Do you understand what I'm saying there? Okay. So this goes back then to the prevention is better than cure angle. What we're now doing, and this is work led by my colleague Gráinne McAlonan, and I have to say it's kind of the bit of my week when you want to leap out of bed and go, you know, today is going to be a great day in the office. This is just such fun. And so what we do is we take babies who are at risk of developing autism because their mum has already had a baby with autism. And so this baby's chances of developing autism are something like 25 to 30%. And so what we do is we take these at-risk infants and we see, have they got differences in brain biology, and do these brain differences predict their ultimate clinical outcome? Now, the idea here is if we get a predictor that's reliable, it's not that we would target that baby with a pharmacological treatment, it's that we would target that baby with a behavioural intervention, which has been shown to work by our colleagues, both at the IPPN and in Manchester. So we take that baby and we give it an emotional sounds experiment and a non-human sound experiment. And this is what it sounds like. So essentially, the baby would lie in the scanner, it's sleeping, and it would hear something like this, or a pouring water sound, or a mechanical sound. And then we would give them a human sound, like, right, human. If you believe I'm human, that's a human sound. And then we would give them an emotional sound, like, oh, hello, baby, hello. I'm not quite sure what that one is, but you get the concept. Or it's, oh, no, don't do that anymore. So they get a negative or a positive sound, and they get a neutral sound, and we see what's happening to that little baby brain as it's fast asleep. So who here thinks that your baby responds to emotional sounds when it's fast asleep? Yeah, you see? You're all geniuses. I had to get a research grant to prove what you already know, right? I mean, what a crazy world. So yeah, so babies respond to emotional sounds, well, to human sounds when they're asleep, and especially to emotional sounds when they're asleep. And they respond more to human sounds than to non-human sounds in terms of their brain activity. But when we take that group of high-risk infants with autism, there are two differences. Firstly, their brains respond more to non-human sounds than to human sounds. And these are 6-month-old babies. So that's the first thing. They're more hardwired into non-human sounds. And the next thing that happens is that we find that when they do respond to emotions, they're less responsive to emotions than a non-risk baby. So not only do they not respond as well, when they do respond, it's much more blunted. Does everyone understand what I'm saying there? Okay, so that's the first thing. And the second thing is we have very, very pilot evidence that this is a good predictor of those infants which are going to go on to develop ASD. But I stress to you that's pilot evidence. We've only published the very first findings, and we really need to replicate that. But the other thing that we're doing to try and make this of more relevance to some of you, others of you in the room, is to say, well, how true is this also of babies who are born to other kinds of mums? And so we've also been looking at babies who are born to mums who are depressed. And what we found is that those infants born to mothers who are depressed are the opposite of those autistic risk kids. That is, they are hypersensitive to emotional sounds, and particularly negative emotional sounds. So if you've had a mum who's been depressed during pregnancy, her infant's brain responsivity to negative emotional sounds is very significantly greater than the control infant, right? And then the next thing is, well, what potential role might SSRIs play within that? And so what we've compared is, this is work of Michael Craig's. He's compared the babies of depressed mums who were treated with medication during pregnancy and depressed mums who did not take medication during pregnancy. There was no significant difference in the severity of depression during pregnancy that we could detect. No differences between the infants in terms of gestational age, conceptional, etc. And what Mike found was that those babies who were of the mums who were treated with SSRIs were also hyper-responsive to those negative emotional sounds. So we're not quite sure yet what's driving our differences, whether it's the SSRIs, whether it's the mum's kind of biology and stress during pregnancy, or it's a combination of those things. And so that's something that we really, really need to focus down on to see also what impact this has on subsequent development. So that's serotonin, and that's infants. What then is going on in the world of glutamate and GABA? I can feel youth going like, why would I want to know what's going on in glutamate and GABA? Well, to be honest with you, I would have been right there with you until, unfortunately for me, I learned that they are absolutely crucial neurochemicals, the excitatory and inhibitory neurochemicals in your brain, the kind of master clock in many neurochemical systems with very significant downstream effects. And it's crucial that those two systems are in balance for you to have a normally functioning brain. And so what we did was we took a rodent model of autism and we said, does this rodent model of autism have a difference in the glutamatergic and GABAergic system? Number one. Number two, can we reverse that by correcting that genetic abnormality? And also, can we reverse it by using a tool compound, which was given to us by one of our collaborators in industry? So what we found in this rodent model of autism was a significant difference in a particular glutamatergic pathway. We found particular differences in synapse formation, glutamatergic transmission, that we could reverse when we re-expressed that deleted gene. But that's fine, right? But that's a rat. Even I know that, well, apart from some of our colleagues, rats aren't people, right? what we really got to do is to get that into people. I need to know, A, is that ratty model of autism any good? Because if it doesn't recapitulate, in other words, replicate the biology of autism in a real autistic person, it ain't no good. So I've got a great paper and a great journal making little ratties run left rather than right, but does that really translate into real live people? And so for that, what we need to do is to get a technology that we can take and use in that rat or that rodent of whatever type and apply it in the human. So all I'm going to show you now is pilot data, and I absolutely want you to bear in mind this is just proof concept studies. I'm just showing you this in the spirit of openness because we're funded by taxpayers and the EU. But this clearly we're going to need replication, all this work I'm going to show you. So what we've done is we've taken that same rat model and mouse model. We've used this brain imaging technology called spectroscopy. I mean, you don't need to be as sad as me to know what that does, but essentially it gives you measures of glutamate and GABA and other chemicals in your brain. We showed that in that rat model, remember in the dish, we got that difference in glutamate. We show we also get that in vivo in the medial prefrontal cortex and in the basal ganglia. We then say, well, can we find the same thing in humans? And we absolutely do. In those same brain regions, we find the same thing that we find in that genetic rodent model. And not only do we find that, we find a significant relationship between the degree of abnormality and the degree of behavioral abnormality. And we also find a significant relationship between the degree of difference in this glutamate and the difference in your brain functional connectivity. So that gives us a way in. So we've got this thing that's going on in this ratty. We've replicated it in a human. It looks like it's associated with differences in brain functional connectivity and behavior. So now the question is, well, how do we get that into a treatment? And we've taken two concurrent approaches. The first is to work with repurposed compounds. In other words, you take a medicine that's out there already, it's going to be dirty, it's not going to be just hitting the targets you want, but it's the first step in the process. And at the same time, you try and come up with what's called a designer drug. But I'm now just going to show you the progress we're making in repurposing compounds. So we have this abnormality. Then what Grania did was she said, well, what happens if I take riluzole, which is a treatment normally for multiple sclerosis and other disorders. We know it targets the glutamatergic GABAergic system. Can we modulate that difference in EI balance? And if we do, does that modulate the differences in brain functional connectivity? And the answer is, yes, we can modulate that brain difference. And yes, we can partially modulate that difference in brain functional connectivity. So we've now got proof of concept that we can alter what's happening in the brain. And so the next step then for us to get that into the clinic, which we're doing now, is to take it into the clinic, and much like I said to you with the serotonergic approach, the idea will be to take individuals, give them one shot of the medication, see what it does to their functional brain connectivity, and our prediction is that those whose brain responsivity changes will be responders to treatment, whereas those whose brain responsivity doesn't change will not be responders to treatment. So, does everyone understand what I'm saying there? Kind of. A lot of you are probably thinking, like, firstly, why am I listening to this guy? And secondly, none of this is going to do me any good whatsoever in my actual clinical service setting. And so what we're also trying to do is to work out whether we can give you tools, or our colleagues in the pharmaceutical industry tools, that they can use in a clinical setting that isn't just imaging. And so what we know is that there's this particular visual task. It's called a paradoxical motion perception task. You essentially takes 2 minutes, you get them to look at a fine grating on like this, and they say when they can see the line moving or not. And that's really dependent on your glutamate and GABA in your brain. So what we find is significant differences in the performance of that task in individuals with autism as compared to controls. we give them the medication, and we reverse that abnormality in that simple visual task. So again, it's just proof of concept. We need to replicate that, but that makes us increasingly hopeful that we've now not just got a difference in this brain chemical pathway that we can modulate, but that we might have a relatively simple tool that we can then roll out to clinical trial settings and or to clinical settings where it could be used in an everyday way. So back to the same thing, right? I've shown you this difference in this EI balance in the brain, but all that work was in adults. What we really want to be doing, as I said, is stopping that boulder. And I've already shown you the work we're doing in infants. But for me, that's still too late. What I really want to know is what's going on in the womb and what might be driving that. And so, again, this work led by Gronya, we are now carrying out fetal neuroimaging, in which we can measure the brain concentration of that same chemical, glutamate and GABA, that we measure in the adults. And so we are carrying out our work here at 30 to 34 weeks gestation. So the idea is we take this little fellow when he's in the womb, we look what's happening to his brain development in that brain chemical, also his brain functional connectivity, etc. We then look at him as he's freshly laid, as it were. We would then scan him again within the first week of birth, and we'd scan him again at age 3 months, 6 months, and we'd follow him up and to see what happens to him clinically over the next 3 years or so. And this is our really early data. So this is if you are a non-risk, healthy infant. So this is your brain glutamate here. This is in the fetal time point. Neonatal time point, you've just been born. An infant, you're about 4 to 5 months. So you see this reduction, very, very rapid reduction in brain glutamate just after birth, that then starts to pick up again. And I won't bore you with the details, but this is exciting for people like me because there's this thing called the glutamate GABA switch, but you don't need to know about that. So this is your non-high-risk infant. But this is your high-risk infant here as compared to controls. And I apologize we haven't got the confidence bars in here, but believe me, it's significant. So we find the opposite pattern. So what you're hopefully seeing there is a completely different developmental trajectory starting before birth in these very high-risk infants. So then the question, of course, is, well, what's driving that? Is it genetic? Is it environmental? And if it's environmental, is there anything that I can do as a physician to try and reduce that? So, for example, is this being driven by maternal immune activation? Is it being driven by things like stress? Or is it being driven by genetic risk? Or is it being driven by other factors that we need to understand which aren't necessarily genetic? But of course, for us to get any of that into trials and treatments, we need to have the regulators agreeing with us that what we're doing is A, sensible, and B, could be used to help screen compounds and may be used in the context of trials. Because I don't know if you know that for the EMA or the FDA to approve a psychiatric drug takes about 50% longer and is about 80% more expensive than for a non-psychiatric drug. And most trials fail. So can we persuade the EMA or the FDA to start accepting our approaches for biomarkers and stratification in trials? And the answer is yes. We've come up with a joint publication with the EMA in which they are accepting our approaches and which they've given guidance as to how they can now be brought into clinical trials in the real-world clinical setting. And so to do that, we've now established a clinical trials network which is across Europe, in which we will be running trials of compounds, both repurposed and novel. But we're going to not be doing it in the usual way. We're going to be using these tools to try and identify much more homogeneous groups of people in whom we will target our interventions at. That's number one. And number two, we won't just be examining pharmacologic interventions, we'll also be seeing how these combine with improving the efficacy or not of other, say, educational interventions or family interventions or psychological interventions. So this is where we started, tins of soup. I hope I've persuaded you that we are not there yet, but that I think we are beginning to make progress to be able to define much more one tin of soup, much more being able to pass out that biological variability into homogeneity, so that we can much better target our individualized treatment approaches, which is just a scientific way of saying what you already know, actually, as clinicians, right? Like, once you've seen one person with autism, you've seen one person with autism, or every person is different. You know that you individualize your treatment approaches currently to the patient that's sitting in front of you. This is just taking it a step further and individualizing it much more around specifically the biology. And when I say the biology, I don't want you to think that this just means brain imaging. It could be subsets of performance on a cognitive task. It could be subsets of performance on that visual task I've given you. So it's not just highfalutin brain imaging. And again, what I'm showing you, hopefully, is the need to bring all these approaches together, the need to think across the lifespan, and the need to translate our work back into rodent models and to forward translate it from rodent models into humans. If you want to have more information about what I've said, please take a look at this publication that's up there, and that will let you know in a little bit more detail. With that, I'll finish, and thank you for your attention. Sure.
Thank you very much, Declan. And I have got time for some questions, so I've got a couple of burning questions. But do you want to just make yourself... Oh, here's a microphone coming.
First of all, thank you very much. Fascinating. I was very struck by the change in the brain imaging with serotonin reduction. Were there any clinical observations?
Yeah. No, there weren't, because that was a one-day trial. So you'd come in, you'd have placebo on one day, and you would then the next day get your reduction in serotonin. We did ask them to self-rate how anxious or depressed they were feeling, etc., but we didn't get any impact on that. So I genuinely don't know what impact that would have on symptoms.
Just thinking about that, Declan, I mean, tianeptine's obviously a very good idea. I can say that because it was mine. But there's 14 different postsynaptic serotonin receptors, so there's the possibility of actually blocking the effects of serotonin quite exquisitely, actually. I mean, that's something that might be worth following up, really.
Oh, totally. Yeah, totally. The question, though, is what's out there that's available that you can rapidly repurpose?
Well, actually, I mean, we have some interesting drugs like vortioxetine that I'm talking about. It's got about 4 or 5 different postsynaptic antagonist effects, so it might be worth talking about that.
Yeah, absolutely.
Any other questions? Question behind you there, just behind you.
I thought mirror neurons were quite important in this area in terms of sort of social interaction. Is that something you've looked at all?
Yes. So the point that's just been brought up there is that one of our colleagues, particularly in Scotland, is very fond of a thing called the mirror neuron system, which essentially is this. Got it. So he eventually smiled. So it's you kind of mirroring what I'm doing, and it's the brain neural system that underpins that. And it's said that there are abnormalities in the mirror neuron system within individuals with autism. That's a hard thing to prove because there have been no post-mortem studies which have really addressed that directly. But we know the whole system looks like it functions differently. But I'm not sure quite where that takes us, if you see what I mean, because we don't have a specific medication for mirror neurons or the mirror neuron system, but yeah, that theory is still out there. Yeah.
Question down the front, Frank. There's a microphone coming, if you just...
Thank you very much. Fascinating. The one result that is extraordinary in terms of treatment of autism that's come out in recent years is oxytocin, which seems to make some people with autism more aware of things like facial expression. How does that fit in with your concepts of the neurotransmitters in the brain?
Okay. So the question was about oxytocin, which I deliberately avoided today because there just wasn't time. It's a slightly controversial area, slightly controversial area in that it makes sense. We like oxytocin. It's clearly to do with social bonding, etc., and there's really good animal literature around it. The early trials of oxytocin looked great, but the subsequent trials have all failed. And we don't know why that is, whether it's because of the things that I was telling you about, which is that we're all looking at different populations of individuals. I can tell you that I think that there will be some really, really exciting stuff released in about 6 months or so in that area, but that's not published yet. In a particular subtype of the oxytocin receptor, I think there's something that's really hot that might take off. But again, I'd stress to you this. If you hear something in the next few months, you probably will, about this amazing new potential treatment coming forward, it's absolutely not going to work in everybody. The key will be identifying those individuals who may respond.
Lady there with her hand up. There's a microphone just coming.
Hi. So what you've said is that there are spectrum disorders and we have to homogenize research groups. But isn't that true in psychiatry, in depression, in schizophrenia? It's almost like we are facing the same problem in most of the diagnostic categories. Would you want to comment on that?
No, absolutely. You know, it's a shame, isn't it, in some ways that all your training is around are they schizophrenic or not, or are they depressed or not? But actually, it's a bit of a nonsense because one of the things we do know is that symptom domains are transdiagnostic. And so one of the things we maybe want to be concentrating on more is, well, actually, what drives executive function abnormalities and how is that different across these different disorders? And what can we learn, say, about executive function abnormalities in disorder X that we can apply in disorder Y? And is there a transdiagnostic treatment approach? Does that make sense to you? But what I'm saying about, you're quite right, what I'm saying about autism is absolutely not true just of autism. It's absolutely true of every other disorder pretty much you can think of.
So, lady in the middle there with her hand up, that will be the last question apart from my question, which I'll...
Thank you very much. I just wanted to know if there are any clinical phenotypes linked to these neurochemical abnormalities like 5-HT and GABA-glutamate and oxytocin. Clinically, is it possible in the clinic to identify these groups?
Yeah. So did everyone hear that question? Is there any clinical phenotype that maps onto these differences in neurochemistry or the brain phenotype, right? For those of you who aren't aware, I actually paid that person to ask that question. Okay. Because the answer is we don't know, but hopefully we will know because we're just now at the first wave of analysing our data from that European study. So we'll have data from 1,000 individuals in whom we'll be able to address exactly that question because we'll be sufficiently powered to do it. In other words, our population size will be large enough so that we can address that question that you're bringing up, and that's a crucial question.
So last question from me, and you had some lovely slides, Declan, some of which I look forward to using in future talks myself, but one of them was the elephant in the room.
Yeah.
And you sketched out really a very collaborative approach between clinical and basic science. It was all across Europe. How's Brexit going to affect that? I mean, you've got a great machine there. Is it going to be knocked off the rails, or I mean, what's your view about it? What do you think we should be advocating for to the British government to make sure that this isn't endangered?
Yeah. Well, look. Oh gosh. Look, we're all psychiatrists, so I can start with a confession, right? My own mother voted for Brexit. Can you believe it? It's an extraordinary woman. I always thought she was extraordinary, but unbelievable, right? How is Brexit going to affect us? It's going to affect us very significantly on a number of levels. Number one, our ability to attract in the best PhD students. Number two, our ability to attract in really bright colleagues, clinical colleagues from across Europe to come and work easily within the United Kingdom. That's going to really very significantly impact upon us. And it's potentially going to impact upon our ability to share data. So that, for example, there are now European laws that govern things like confidentiality, et cetera, which are going to be difficult to imagine how we'll deal with the new era. And of course, potentially, it will impact on our ability to carry out large-scale research. But apart from that, it's all good.
But not everywhere on your map was part of the EU. So I mean, it might be possible to square this circle because you have partners that are outside the current EU, don't you?
We do have partners that are outside the current EU, yep. So if you're suggesting, Alan, that in future I'll be fine because I could collaborate with the Ukraine, Montenegro, Lithuania, and Fiji. I don't know if anyone's from those countries here. They're wonderful countries, but yeah, a bit more tricky.
Okay, so we need to be writing letters to Theresa, I think.
Yeah.
Okay, well, thank you very much. I'm just going to do a brief vote of thanks, and we always ask the keynote lecturer to give us a state of the art of their chosen field, and we've heard that not only in terms of autism and the underlying biology and how that may translate into development of new treatments, but also really a textbook example of how to examine, to organize big science in terms of mental health. So I'd like to ask you all to join me once again in thanking Professor Murphy for an outstanding talk.